this video is going to focus on the first type of immunity, which is cellular immunity, and then there'll be a second video about humoral. So cellular or cell-mediated immunity is the type of immunity, remember now we're at the specific defense, the type of immunity which is forming a specific defense from T lymphocytes that directly attack and destroy diseased or foreign cells. So it is a cell that is directly attacking another cell. Okay, so that's easy to remember that it's cellular immunity. So we are talking about intracellular, that has to do with viruses or bacteria or foreign cells. The immune system is remembering the antigens and prevents them from causing disease in the future. So part of this immunity definitely includes the memory component. Okay, so we're going to talk about 4 types of T cells, and some of them are more important than others, but we'll go through the types and then we'll see what each of them is going to do. So cytotoxic T cells are the effectors of this type of immunity. They are actually the ones carrying out the attack on the enemy cells. Don't worry about all of those proteins over in the parentheses. I'm not worried about that. The helper T cells help the cytotoxic T cells, and then they also help the B cells kind of jump into action, and then also help with the non-specific resistance that we talked about previously. They also have regulatory T cells, which are sometimes called Tregs. They stop the multiplication and secretion by other T cells, which limits the immune response, and we'll see when those come into play. And then the memory T cells, they have to do with responsible for the memory of the cellular immunity. So those are specifically the cells that are remembering which pathogens have been in your body before. Okay, so for each type of immunity, both cellular and humoral, we're going to follow through with these same 3 stages: recognition, attack, and memory. So for recognition, the first step, the antigens have to be recognized, and they have to be presented in order to be recognized. So remember we talked about these antigen-presenting cells, or APC. They encounter an antigen, and then they basically migrate to the closest lymph node and display it or show it to the T cells. When the T cells are encountering that antigen that's been displayed, then that is when they initiate this immune response. This talks about 2 types of proteins that can be displayed, or 2 types of antigens. The T cells are going to respond in different ways. So the first one is your MHC1 protein. In the previous videos we talked about this as a self-antigen. This occurs on every nucleated cell. So all of your cells, they are carrying this little protein on the outside of their cell membrane. The other type is the MHC2 protein, and they only occur on these APC cells and on foreign antigens. So the T cells, or the cytotoxic T cells, only respond to the first, that self-antigen, and then the helper cells respond only to the second one. So when the T cells are activated, they bind to that protein, and then they also have to bind to another APC protein to basically check twice. They want to check twice, am I really bound to a foreign antigen? And that process is called co-stimulation. It's just kind of a backup. Are you sure this is a foreign antigen? Okay, yes. Okay, now I'm going to start my response. So if that co-stimulation, or that second check of the antigen, is true, that is a true foreign antigen, that's going to trigger what's called clonal selection. And so at that point, the T cells are going to copy themselves. They're going to undergo repeated mitosis, and to make a clone of that identical T cells. Remember, these T cells are specific to the type of pathogen. So once those T cells are cloned, then they can become effector cells and actually carry out the attack. Others are going to become memory cells. Okay, so this gives you a visual of what's happening. And so the T cell is going to bind to that APC cell and that foreign antigen. And then the second image is showing it kind of double checks on another protein. Is that really a foreign antigen? Yes. At that point, the T cells multiply themselves, and then it shows if it's either becoming a C T cell or a helper T cell, what the response is after that. I would encourage you to watch through these animations. So after the recognition, the attack is the next part, and so the helper T cells and the cytotoxic T cells are going to play different roles in the attack phase. So the helper T cells are necessary, but they kind of coordinate this cellular immunity, and they recognize that antigen. They secrete these interleukins, which kind of make this whole process happen. They attract the neutrophils, the NK cells, the macrophages, and then they also stimulate the T and B cell mitosis. So they're not specifically attacking themselves, but the way I remember it is they kind of provide backup. Whereas the cytotoxic T cells are the actual T cells that are directly attacking other cells. And so when they recognize that foreign antigen or a diseased cell, they are going to dock on that cell, secrete chemicals, and then destroy that cell and move on. It's very similar to the process of those NK cells that we saw during the non-specific resistance. They make a hole in that cell membrane and then have their enzymes go in and then break apart the enzymes of the foreign cell. Again, I keep using the word foreign. I guess what I should be saying is an infected cell, a cancer cell, or a foreign cell. Okay, sorry, I'm getting a little ahead of myself. So this talks about the kills the cells in the same manner as those NK cells. So the perforin was that protein that makes a hole in the cell membrane, and then the granzymes are the enzymes that go inside to destroy the enzymes of the host cell. And that's what happens. So that's how the T cells are going to attack, the same process that we saw in the NK cells. Okay, and then the last step of cellular immunity is memory. So after the attack, the immune memory comes next. And so we said during the clonal selection of those T cells, some of them are going to become memory cells. Those cells are long-lived. They are going to keep that memory of what that antigen was for their lifetime. And they're more numerous than other types of T cells. And they don't need as many steps to be activated, so their response is quicker. So if you are re-exposed to the same pathogen later in life, those memory cells launch into a quick attack, and potentially you wouldn't even notice anything if they jump into response quick enough, you wouldn't even notice any illness.